Hello, Philip. We'll be starting in more 10 minutes, rather 7 minutes. So, thank you for waiting. I welcome you. Hello, everyone. Thank you for joining today's webinar. We'll be starting in more 5 minutes. We are waiting for other participants to join as well. Thank you for joining. And meanwhile, we begin with today's session. It would be nice if you could please introduce yourself in the chat box and let us know about your educational background and your research interests. A very warm good morning, good afternoon, or good evening in whichever part of the world you are. I would like to welcome you all for today's webinar on OmicsLogic, Bioinformatics, and Data Science Research Internship or the Fellowship Program. The Research Fellowship Program has been designed to help young researchers and students take advantage of the bioinformatics resources for analysis of complex life science data and become versed in bioinformatics. The research fellows participate in cutting edge bioinformatics research, which is led by expert mentors. The fellowship program will offer a combination of online resources and mentor guidance to prepare you and help you complete a bioinformatics project. The registration fees would cover the cost of training, mentorship, and big data T-BioInfo Server. And the platform is a cloud-based analytical server which is used by the research labs and independent scientists around the world. Today, we'll discuss about the research specialization track, the program structure, outcomes of the program, and take your queries and listen to our Research Fellows project. We will also hear from two of our Amity Research Fellows, Mr. Hriday Thakur and Ms. Akanksha Singh, about their research projects in oncology and neurosciences. So before we begin with today's webinar, let me introduce us. We are a US-based bioinformatics company who is working with multiple academic and commercial collaborators to develop easy-to-use analytical tools, and our mission is to make bioinformatics more accessible. I would like to introduce myself. I'm Sonalika Ray, and I'm the OmicsLogic Community Manager, and I'm responsible for daily interaction with thousands of our users that are a part of our bioinformatics community, which is managed by Pine Biotech. Over the past several years, we have had the privilege of working with a number of organizations developing a training program for research-oriented students, faculty, and democratizing bioinformatics, making this discipline accessible to thousands of students and faculty around the world. The OmicsLogic research-based training programs are a result of close collaboration between Pine Biotech and numerous academic institutions that have participated in content evaluation, curriculum review, and project design. The training has been completed by over 24,000 participants from 187 countries in over 300 workshops. And due to this fast growth, our team is working with local and regional coordinators that are helping refine the local program logistics and leverage our online training resources, adapting them to the needs of students and researchers around the world. The programs follow a project-based learning approach, which uses research-grade tools to analyze data from top peer-reviewed journals. The training has been completed by the participants in the fields of oncology, infectious diseases, precision medicine, neurosciences, data science for biomedical data, and comprehensive training on omics data analysis. As a result, we have acquired professional certification to offer our curriculum and support research projects using our OmicsLogic project-based curriculum. And together with our existing modules, for which you will gain access to the OmicsLogic Learn portal, where you can try and explore these modules, exercise in pipeline building, coding, and various methods of analysis using project sources for peer-reviewed publications. All right, so now let's highlight some of the current challenges and limitations and how we at OmicsLogic help resolve these with our training and research services. So what are some of the challenges that the field of bioinformatics is currently facing? To begin, this field is constantly growing and developing new tech and software to improve processing time and power. The data sets that are analyzed are usually very large, and they take immense amounts of computing power and memory that many personal computers do not have. Also, despite being publicly available, data is usually complex to access, and coding knowledge is usually required to do so. Similarly, analysis pipelines and programs require coding knowledge that many biologists do not have. In a university setting, students have the opportunity to learn the background information of designing and implementing a bioinformatics project, but do not receive practical hands-on experience with the full workflow due to limitations mentioned previously. Similarly, faculty faces challenges with being able to cover a diverse range of topics if they are specialized in a certain area of bioinformatics or if they are unfamiliar with bioinformatics in general. For example, a professor whose research focuses on oncogene expression might not much know about metagenomics of seawater. So to overcome these challenges, the OmicsLogic platform was developed. This program aims to standardize, organize, and systematize bioinformatics training in order to make these analyses usable for any biologist or clinician. Some problems for standardization includes materials for these courses are usually developed in-house, taught sporadically, and are rarely updated. Organization, because the challenge is to combine several different topics together. The flow and transitions are needed to be developed. And systematization, how can you know that someone is stuck and needs help or allow someone to run forward? OmicsLogic uses standardized modules and interactive sessions beginning with mastering terminology and concept to implementing these concepts with real data sets in a project-based approach. So, what is the learning process? In order to create a clear path of skill development, OmicsLogic employs the Bloom's taxonomy levels of learning. This approach starts with the basics, where students recall facts and terminology and are able to repeat and memorize the material. With this base, the learning builds to conceptual knowledge, being able to understand concepts and apply information to new students. The student will then be able to analyze data by drawing connections and evaluate when to use certain methods. This leads them to the highest level of learning, the ability to design and create original work. Also, we will take you through the program resources today and help you get started with your training and research journey. The Research Fellowship Program will also give you an access to the OmicsLogic courses, which includes courses on bioinformatics, precision medicine, multi-omics, including genomics, transcriptomics, metagenomics, and epigenomics. Data science that includes bioinformatics using R and Python, and example projects of different fields which you could learn from and replicate for your research work. The users will also be given directions to access the T-BioInfo platform for bioinformatics processing and analysis of transcriptomics data. The platform includes demo pipelines as well as data management and analysis cloud infrastructure to run bioinformatics pipelines. Different stages of analysis are performed in different sections of this multi-omics platform. So first of all, now to proceed ahead, let's begin by signing up on the OmicsLogic Learn portal. And once you have signed up, please make sure that you have a complete profile, which will include a profile picture, full name with appropriate capitalization, link to your social media handles that includes Facebook, LinkedIn, Instagram, Orchid, and Twitter, and a brief bio about yourself that will consist of educational background and research interest. The mentors can view your profile and your progress under the Activity tab. The courses you are currently completing will appear under the Courses tab. And finally, after the completion of your course, the certificates will appear under the Certificates tab. So that was it that I wanted to tell you about us, what we do, and how you can get started with your research journey. Now let's hear from our research fellows and look what their research works were all about. So I would like to invite Mr. Hriday Thakur from Amity University, Noida. He's a student of BTech Biotechnology and has just finished his dissertation with us. And that's it about him. I would now like to pass on the stage to Mr. Hriday. Are you here with us?
Hello, I'm audible, Sonalika.
Yes, Hriday. Let me give you the access. All right. Can you share your screen?
Yes, just a minute.
Yes, Hriday. Just a confirmation, we can see your screen.
All right, thank you. Is the first slide visible where we
Yes,
have?
it is.
All right, thank you so much. All right, hello everyone. Hope you're doing great. Just a minute, my laptop is malfunctioning. Don't worry. All right, fine. So, thank you, Sonalika, for inviting me, first of all, and to everyone who's present in the meeting and who wants to understand and know what we did. Here is the presentation of the same. Now, first of all, I would like to share my experience. Ki what we did at Pine Biotech was really immensely meaningful to us because we understood the entire analysis part of how you can really perform the analysis on biological data, because it was something that is very new to us, but we still understood in the given time frame of about 6 months that we had for a dissertation. It speaks volumes about the organization itself. So let's start with the presentation. As you all can see on the screen, my presentation and my topic was identification of gene signatures associated with EGFR mutant cancers, and we had to understand the role of vitamin D in their treatment, right? So the two more important things in this is that we took breast cancer first of all, and then we took the EGFR mutant cancer specifically, and the third most important thing is the role of vitamin D in that, in the treatment of breast cancer. All right. So first of all, we have to understand that living in India, we have a very high prevalence of deficiency of vitamin D in both males and females. Now, this can be attributed to lifestyle changes. This can be attributed to difference in or very lack of physical activity or the sunshine that we get. But still, the end result is that we have a very high prevalent population of deficient vitamin D or deficiency in vitamin D. Now, this vitamin D deficiency was also seen in US about in the 19th century or so. And what they did, there was a publishing that happened that correlated the low, the deficiency in vitamin D with the high prevalence of breast cancers. Now, this study was very interesting because this is what we had based our research on, and the end result was, of course, could be anything from recommending or increasing the recommended dietary allowance for vitamin D to actually finding out a novel biomarker for vitamin D in breast cancer. All right, moving ahead. Now we have to understand the effects of vitamin D and its effects on the body, but I'm sure this is the biology aspect of it, and it's a little bit complex. But to understand is that the breast cancer cells have vitamin D receptors. Now, when we say that breast cancer cells has vitamin D receptor, that means that when vitamin D is absorbed in the body, then it will be broken down into the breast cancer cells as well. So that means that there can be a correlation in the cell cycles in the entire metabolic range of vitamin D, where it can affect breast cancer. So there's a big can that we wanted to figure out and understand whether it's useful for us or not. All right, this is the methodology that we followed. The first and the most important part was selecting the data sets. Now, I would say that this was the most important and the most complex task that we had to go through, because one, it is tough to find data sets combining both vitamin D and breast cancer, because first, what happens is that there are not many publications that is done. Second is that whatever publications we had or were found were very old or very new, consisting of very few sample size. So this was a very tough task, but Raghavendran sir really helped us in this. So finally, when we found the data set and we got the data set, we had to perform the analysis on it. Now, before that, we had to clean the data. We had to analyze and understand some basic aspect of the data, series matrix. So from that, we did an exploratory analysis, and after that, we did a differential analysis on the data. Finally, what we did, we understood the data analysis that happened, and from that analysis, we drew a conclusion, that is on point number 3, is whether we can find common genes in the data set and we can understand their function or not. right? So I'll take you to the data set right now. All right. So in a crux, what happened is that we found two data sets that were very appealing to us. There was one lung adenocarcinoma data set. This lung adenocarcinoma was treated with the normal treatment of cancer, the lung adenocarcinoma, the ositinib treatment that is given. And then we had the triple negative breast cancer cell line that was treated with normal treatment and secondary a cofactor, a dual inhibitor, that is PHA 767491. Now, the common thing in both of them that both of these were EGFR resistant. So we had a common ground. We had a different treatment method. If the triple negative breast cancer cells had the same genes that were upregulated, differentially expressed in the lung adenocarcinoma, then we could say that we have a result on our hands. Also, if it's not the same, then we can also conclude that the tissue matters, the tissue anatomy or morphology matters in the case of vitamin D and its effects on cancer, right? So moving ahead, so this is the result now I'm sharing with you. When the breast cancer was treated with both the treatment, that is the laptinipib, and PHA treatment, that is dual CD7 CDK9 inhibitor, these were the genes that were differentially expressed, right? And to know more about these genes, you can of course search on Google to know more about these genes, but these genes we found really interesting. So I have just noted them down for understanding. For example, that MEPCE gene is a capping enzyme, right? It produces a capping enzyme. Now, there are studies which tell us that MEPC depletion affects the expression of cancer relevant cases. Now, hence, it is important for us to understand the role of this MEPCE gene. Second was caspase. Caspase 7 again is a very common gene that is found. Now, in some studies, again, it is said that it is aberrantly expressed in breast cancer cases and it can cause cell growth and proliferation and downregulation of a protein that Cas7 positive breast cancers could be one of the potential therapeutic strategies. So decreasing the amount of caspase-7 can really help us in understanding or curing breast cancer. The next is SN1P1 gene, that is SMAD nuclear interacting protein. Again, it is a very common gene, but this gene was important in breast cancer, and again we can say that lung metastasis of breast cancer. Hence, we took out this gene specifically. Moving ahead, for the lung adenocarcinoma data set, these are the three volcano plots we found. Now, you have to understand why three is because we had to study the effect of only the drug, only vitamin D, and when both vitamin D and the drug were given. So this is only vitamin D, this is only the drug, and this is both drug and vitamin D. As you can see, both of these genes are overly expressed or differentially expressed in the third one, right? So this is what we needed. This is what we wanted. The CYP24A1 gene is something that we could positively look at in terms of effect of vitamin D on breast cancer. And although it's a very less result to be found, or a very but it is something. So we're happy that we finally got something of interest. So what we have to understand is that CYP24A1 gene is a very common gene, right? Because it is found in the cytochrome P40 system in the liver. But this is present wherever vitamin D breakdown needs to be taken place. So what we concluded is that whenever there is an increase in the level of CYP24A1 gene, there is a potential that it can be used as a biomarker, especially in predicting prognosis. So predicting and prognosis purpose, it can be used. So this was something that we proposed in the result. Ki, yes, if this gene is overly expressed in vitamin D cases, then it could be used as a predicting biomarker for breast cancer. The only thing that needs to be done is in the next slide, what we can do, right? So we found that vitamin D supplementation can really elevate inhibitor resistance as seen in the breast cancer treatment. We found that it is happening. Now, we also found that the vitamin D treatment needs to be further investigated for other cancer subtypes and other cancers as well. And if vitamin D can assist the individuals with breast cancer to control the symptoms and live longer, you have to understand what you will have in your hands if this study is proven. It's like a David and a Goliath story where a simple molecule as vitamin D, if proven, can fight breast cancer, then you are fighting the entire pharma lobby, because they are using millions of dollars of drugs to fight these cancers, and you will be putting a dent on their profits big time. So if this study, anybody who wants to take up this study, can prove that vitamin D can really affect the treatment of breast cancer and can help in treatment of any cancer for that matter, I'm saying really can prove, then they have a huge fight on their hands. And if you really want to watch the fight, I suggest you watch this movie called Concussion on Netflix, just out of interest. All right. So moving on, the further studies that can be done in this prospect are, there's a vertical possibility of studying this, because you can look deeper into the effects of genes on breast cancer. So vitamin D effects of other genes of breast cancer, you can do that. Secondly, what you can do is horizontally you can study different cancers and effects of vitamin D. And if there's a correlation, then you can put that correlation in that yes, there are different cancers which have similar effect when they are treated with vitamin D. So this can also be a very good topic to understand. So finally, this is because I was presenting this for my college university as well, my university as well. So I had some articles that told us the effect of vitamin D on cancer. These are pretty recent studies that are done. So as you can see that people are going in this direction, that there are therapeutic benefits with different types of cancer. You have leukemia here, you have different cancers, colorectal cancer as well. So people are going in this direction to understand that vitamin D can really be a miracle molecule for helping out and fighting cancer. And with that, thank you everyone. Let's use the science and let's use what we have learned and the knowledge to cure the world. Because I will finally just say this key, there should be a usage to what you learned in classes. It's important to, yes, study, but it's more important to apply that study somewhere. So we gave you a direction in this that yes, vitamin D can really help us fight cancer, but it is up to you to take up this fight further and finally prove to this world that yes, it's a miracle molecule and it can be used. Yes or no, or whatever the results might be, but you have to take this fight to help out the people who are fighting this menace in this world. So that's it. Over to Sonalika. Thank you so much, everyone.
Thank you so much, Hriday. That was a wonderful presentation. I really like the way you presented your research. And yes, exactly, Sparsh, the way you explained and how you have put together all your results together. And now I would like to hear from Dr. Raghav. What does he think about your project and how further you can improve, maybe if there is any scope. Over to you, Dr. Raghav.
Thank you, Sonalika, and thank you, Hriday. That was very well presented and very. It has the overtones of a lot of popular lectures, and thank you for taking the research beyond answering a scientific question's point of view.
Thank you so much, sir. Thank you so much.
Sometimes, or most of the times, when we are working or when we are focusing on a scientific query, the implications and the real world applications and the enormous potential of that might not be our focus, and we might be too focused on the research question at hand. So we are all working or using science for the betterment of the world, and thank you for putting that wonderful message forward. Thank you for that. Yeah.
Thank you so much, sir. Thank you so much. Thank you so much, Sonalika, as well. If anyone has any questions, they can post in the chat box, and you can reach out to me on LinkedIn as well. And yes, thank you everyone for patiently listening. Over to you, Sonalika.
Thank you, Hriday, and thank you, Dr. Raghav. Anand wants to ask something. Anand, you can unmute yourself.
Hello, ma'am. And first of all, good evening to all, one and all who is here in this meeting. Actually, I have joined late in this meeting, but I'm sorry, extremely sorry for that because I'm busy in some tuitions and all that. But my question is to you, like how as a I'm an individual, like I'm working as a intern in CSI lab, but I want to also contribute in this project as well. So could you please tell me, as a student of biotechnology, how I can contribute to this project and how it is going to help?
Dr. Raghav, would you like to take that question?
Yes, sure. Can you please one more time repeat as a biotechnology student?
Yes, sure, sure. So actually, I'm a biotechnology student. Right now, I'm pursuing BTech biotechnology. I'm right now in 4th year. So can you please tell me how I can be a part of this project and what you are right now talking, and how it can contribute to the world as well as to me as well for my development in my career as well?
Sure. I mean, Anand, if I'm not mistaken, Anand, biological research is not what it used to be like something like 20 or 15 years ago. 15 years ago, then you will have actually identified some specific questions to answer, and then formulate your hypothesis or questions based on some initial data set or initial understanding of a system. And then you design an experiment, collect data, analyze data, publish the results, or find the results. But where bioinformatics can help in this aspect is due to the availability of an enormous amount of data, it can help you make some important predictions or important suggestions and help you make some critical decisions based on whatever data that is available. Like you might want to focus on a species, and a very closely related species might have been investigated in a greater detail. So you can look at that specific publication and that specific data and design your experiments and try to understand your question from that information. So these are the ways in which you can employ bioinformatics as a supportive science for main or your research in focus, which could be based purely on biotechnological protocols. But bioinformatics as such, as what we are trying to do is we are trying to use or leverage publicly available data and trying to understand the pattern that is available in not one, not two, but many more of these data available, and trying to make some conclusions for the biological question that we are trying to address, or for the research area that we are trying to focus. As you also know, there is enormous amount of data that is available. So our science depends on this availability of data and good quality data. And most of the time, we will actually spend very careful amount of time in picking the right data that will help us understand the question. So if you are interested in expanding your horizons beyond your lab environment, like using bioinformatics methods to aid your research, or you can use bioinformatics methods to completely focus on the problem from a different dimensions, right? You don't have to generate any data, but you can use whatever data available to find some important and critical results and implications and inferences from your data. So these are the ways in which you can make use of the research fellowship program to help yourself. So in a nutshell, you can either learn the nuts and bolts of all the analysis methods and the different comparisons of these results and the different comparisons of the sequence of these analysis methods for different omics data type, so that you can expand your horizons or expand your skill sets. Or you can take part of your long-term project and do a bioinformatics based analysis on that specific part, so that it helps you in the long-term future. So it depends on what you want to know, what you want to do, and we will help you in actually finalizing and in understanding what you are clearly interested in. So we will take it from there and give you pointers in trying to show you directions in which way you can go to achieve what you are trying to understand or what you are trying to investigate. Okay.
Yes, sir. I understood well.
I will then pass the stage back to Sonalika.
Yes, thank you so much, sir. And are there any more questions? You can feel free to please unmute yourself and take it up. All right, so if there are no more questions, we would now like to address a question on our behalf that we usually get asked. That's about various specialization tracks. So, what are the different specialization tracks, or what are the different data analysis types that a researcher can work on? And before working in a specific field with us, you need to get yourself trained. So, what are these fields or types of data analysis courses that you can take and learn from and then get started with your research project? So, for that, we have a video recorded by a mentor, Dr. Raghavendran. And I would now like to request Ujasvi to please share his screen and play that video. After the video recording is over, the participants can unmute themselves and take their questions. Dr. Raghav is here with us. He will answer them. Thank you so much. Ujasvi, can you? Yeah. Okay. Thank you.
So this is a consolidated web page for what the research topics and what the research fellowship program is all about. So when we go down, I think you would have gotten a lot of introduction about how different students and researchers can make use of big data while performing different analysis. And what do I mean by that? Let's go down. You will get these specific advantages of when you sign up for the research fellowship program. So you have curriculum that's specifically designed for your research area and for your research interests, and you have your own self-paced learning, and most of the courses and most of the resources that we are going to share with you in the research fellowship program is asynchronous, meaning you will learn at your pace, you will do this at your pace, and then we will be there to discuss with you about your progress and about how to proceed further in the research fellowship program. And you will get hands-on with big data as expertly demonstrated by 3 students' research topics. So all of them obtain data from publicly available forum, and all of these data are big data, big omics data, and they used this big omics data to address different parts of the biological queries and questions. And you will have access to expert mentors like myself, like Dr. Mohit and others. So when you start learning, you will get immediate feedback from us, and we are accessible to you offline. And also we will meet you once or twice during the week, during the scheduled research fellowship meeting. And also if you request for one-on-one meeting for some specific cases, then we will offer that also. So when you go down, what are the different areas in which the omics data and multi-omics data in general are being used to address specific research questions? You have a variety of topics that uses omics data as of now, which will include genomics data, transcriptomics data, epigenomics, metagenomics, and these are only few that we are targeting or that we are actually focusing on. You have furthermore, you'll have proteomics, you have metabolomics, and etc. A lot of other omics data types are coming up in trying to address different areas of biological research. So, and we have actually this site, this web link gives you some of the major applications of omics data analysis to different areas of analysis that is described, that is focused. So some of the obvious focuses on infectious diseases, and the pandemic era that we are currently living is a perfect example for what omics data can achieve in addressing infectious diseases case. And we all know different terminologies related to SARS, like variants of concern, variants of interest. How does it spread? Like what are the different lineages, right? and what are the mutations that actually characterize variants of concern, these variants of interest. And all of these analyses are direct results from genomics data and transcriptomics data analysis. So when you use the whole genome sequence of the virus and try to compare it with the other strains and other variants of this same virus, you will get a lot of information. And this is just one example of what you can achieve from sequencing the whole genome sequence of the virus. And similarly, this can be extended to transcriptomics landscape analysis, which will characterize how a patient is going to respond to the onset of the disease or to some drugs that is going to fight against the progression of this disease. And these are just two omics data types that is trying to provide us with a lot of information in addressing some of the key questions in infectious diseases. And we can again do this in metagenomics data analysis and epigenomics data analysis, which are also extremely important omics data types that are coming up. And I would like to stress the importance of metagenomics data analysis, where the gut microbiome or the microbiome estimation of the human gut is being linked to various other disease onsets, like cancer, like allergy, like even neurological disorders and pathological states. So that level of association is being exploration of that level of association is being carried out at various different labs, and this is one of the interesting and upcoming omics data types analysis methods and the questions that we can answer through analyzing and observing patterns within the metagenomics data. So when you go down, we have a lot of project examples on Omics Learn, which focuses on different omics data analysis in trying to address key questions in infectious diseases like SARS, like malarial infection, like the SF9 cells will actually tell you about how to identify viral contamination in your samples. And Ebola virus epidemic is an excellent example of how genomics data was used to track the progression, disease progression and spread of Ebola virus. So when you come down, the application of omics data analysis is at its peak to oncology because of the requirement and because of how omics data characterizes tumor, cancer, and generally the disease progression and its effect on various types of patients and various types of various types of races and ethnicity. So what we get, part of the reason why it is required, because of the existing traditional methods of identifying, characterizing, understanding the disease progression of tumor and treating the disease, tumor disease and cancer as such, is not sufficiently effective. So hence we had to resort to transcriptomics or genomics data analysis, epigenomics data analysis, and etc. So when we have a wealthy of such information and big data, it is natural that methods that are specifically designed to analyze patterns from these large quantities of data, like advanced statistical methods involving artificial intelligence, deep learning, machine learning, will come in handy. So they are also at its most useful when you are trying to use a large volume of data like transcriptomics data from lot of patients as compiled in a wonderful public repository like the Cancer Genome Atlas or TCGA. So we also have a NCBI database where the SRA data will provide you a lot of genomics information and genomics data where you can focus on, for example, genome-wide association studies, targeted loci studies to understand the specific genes and driver genes that are that could be used to answer and that could be used to explore the onset, disease progression, and the treatment response of different tumor from different cases. And there are also efforts in understanding cancer from integrating multiple or multi-omics as such, from maximizing the information and also integrating the omics data type with a wealthy of cancer history, patient history database and patient history data and the treatment information data and genetic data, genetic history data generally that most of the translational approaches will acquire at the time of diagnosis. And we can also use traditional methods like using structural biology and omics data integrated into structural biology for developing different drug targets and drug molecules like chemoinformatics data to uh, to to approach the translational problems of finding and identifying targets for these cancer research, and also finding and developing treatment strategies for targeting these specific targets that we identify from structural biology efforts. As I mentioned in the previous case, metagenomics is one of the upcoming area that has been linked to majority of the onset and progression of disease from various other parts of our body. And this doesn't stop at cancer, and this doesn't stop at gut or chronic diseases development. So again, to give you a few examples of what are the different projects that we have in Omics Learn, we have a project that focuses mainly on the immune response of the cancer to different immune therapies, and we have projects that deals with mass or huge volume of data from the cancer genome atlas to identify risk factors that are associated with liver cancer. And we have a project that actually tries to relate omics data with the treatment response data that is observed from grafting the tumor into model environment like mouse model and like cell line models, etc. And that will straight away lead us from understanding oncology through omics data analysis into translational aspects of oncology in precision medicine through the application of omics data analysis in treating cancer. So for this specific aspect, or for this specific research topic, then we have one project, or we have multiple other examples with this specific project, modeling cancer precision medicine, which I was talking about, where the cancer cells were grown in a cell line where they are grown in cell line environment, and they were treated with multitude of different treatments, and their response to different treatments through increase or decrease in the volume of the tumors are observed. And this treatment response, is directly linked with multiple omics that characterizes these tumor cells. And by linking these omics data type and a treatment response, we can identify which treatments are effective and how we can use omics data types to effectively or to accurately identify the effectiveness of this treatment. And this will fall straight into patient stratification in terms of which patients or what are the subset of patients that will respond to a specific treatment, and treatment stratification in terms of what are the possible treatment options that I can suggest to a patient who is likely to respond to several different treatment options. And this doesn't stop at treating hosts from human or from majority focus on Homo sapiens and human. This also extends to agriculture, where I believe the analysis has a lot to offer, and the application has a lot to offer in terms of understanding, for example, drought resistance, understanding, for example, salinity resistance, understanding, for example, heat resistance of crops, which naturally have acquired these characteristics since they are being exposed to these harsh conditions. One can characterize these plants using, again, these molecular characterization methods from omics data types analysis, and then try to apply whether bringing about such changes in a different plant can actually result in developing resistance for drought, developing salinity in resistance, and also whether can we transfer the ability of plants to survive in harsh condition by changing the omics landscape of these plants. So such efforts are actually actively going on in agricultural area. And again, to stress on the application of metagenomics data analysis to a wide variety of biology, and in general, people usually take the soil samples, understand the microbiome population of the soil samples from different parts, right? From, for example, from from a region which receives very low rainfall or from a region which receives very high rainfall. So we can understand what are the different bacteria, fungi, and virus that makes use of these conditions and effectively to help the plants grow at its maximum potential. So these are different areas in agriculture which are actively upcoming and developing currently, and both in basic mechanistic research and also in application oriented research in agriculture, in plant biology generally. And neuroscience is also one of the field where people have tried to understand, or people have tried to apply omics data analysis from samples from brain. And there is a practical difficulty in acquiring samples from brain for live patients, and hence now there is a lot of effort in trying to relate
Ojasvi, we can't hear. Okay, it seems that he has lost his connection.
making these connections that are usually not observable using traditional analysis methods, and evaluating them for the statistical verification or statistical significance of these connections. And transcriptomics data analysis, as I mentioned, is one of the most used omics data analysis, or one of the most applied omics data analysis to variety and wide variety of different questions related to human translational biology. And neuroscience is not agnostic to that also. So traditionally, people extract samples once the patients have passed away from the brain cells, and compare it with the healthy patients who are age matched, gender matched, ethnicity matched, so that we are eliminating a lot of other confounding factors in trying to understand the gene expression landscape of neurological disorders compared to healthy patients. So when we extend such analysis to metabolic disorders like allergic reaction to gluten, or like allergic reaction to some specific metabolome, or some people will have acid reflux. So in lot of these disorders, and some people will have difficulty in filtering salts in Sydney. So in lot of these disorders, again, omics data analysis is actually proving to be very helpful, because traditional methods of observing histological data or biochemical assay data is limited in characterizing the tissues and samples in what they offer. And trying to extract the genes, and trying to extract the gene expression data, trying to extract the DNA from within the cell is actually more helpful in trying to understand or trying to get a holistic picture of the samples themselves, which can be useful in relating to various different phenotypical applications and phenotypical characterizes of these samples that we have categorized. Like we can take samples from healthy patients, compare it to samples from who has developed chronic allergy to, for example, peanuts, and we can understand in what are the major ways the transcriptomics landscape differ in between these two samples, depending on where the samples are from. Samples could be from the skin, or from the stomach lining or from the gut microbiome also. So, the various other omics data types are also explored in this biological approach. So all biology is not, I mean, all biology is not related or limited to being applied on Earth alone. So upcoming research in space omics, where NASA and other space agencies from all over the world are heavily focused and heavily investing, is the application of omics data analysis for understanding the effect of space on human body or on model systems. So people have found out effective method to simulate microgravity on model systems and microgravity on human cells themselves. So people observe or people characterize these human cells based on the omics data analysis or molecular characterization that will take multiple different omics data characterization, and then compare the data landscape before and after subjecting the samples or subjecting the model systems to microgravity or to space travel in terms of astronauts, or in terms of even they took mouse to International Space Station once to perform these analysis. So these are the different fields that biology and bioinformatics and omics data, big data, omics data analysis are actually influencing actively and very rapidly, right? I mean, space omics is kind of a field that nobody would have heard about 3 years before, and right now it's actually picking up, and even a lot of upcoming and developing countries are actually focused on performing or understanding the effects of microgravity and space travel on astronauts and other subjects. So we have these different fields that are being actively explored on by students as they have presented you their research topic of interest. So it depends upon what you want to focus on. For example, you can focus on cystic fibrosis, you can focus on SARS-CoV-2 characterization using different analysis method. You can focus on long non-coding RNAs for being targets in leukemia, and you can also focus on different trying to understand biomarkers for prostate cancer or cancer types that is not being studied actively, like cervical cancers, prostate cancer, and etc. So this doesn't stop at the framework or at different biological aspects that we are limiting you to. You can also trying to mix match these analysis methods and different biological questions that you can answer from this. Look or explore this page, and this web page and web link has lot of information and links forwarded for you to understand the effect of research fellowship program and its reach through the aspects or through the dimensions or through the view of students themselves. You are free to check different submissions from students and the variety of topics that they were interested in exploring omics data analysis and their applications in general. So with that, we also have furthermore links to some of the major or some of the key talks from compiled from our previous webinars and from a symposium that we successfully conducted at the end of, or at the beginning of April rather.
So thank you, Ojasvi, for playing the video. So hello guys, I'm Sparsh and I'm Business Development Associate here at Pine Biotech. And now I'll take you through the registration part and the resources, everything which you'll have access to once you enroll in this program. So let me share my screen. Ojasvi, could you please confirm if you can see my screen?
Yes, sir.
Thank you so much. So this is the program page of Bioinformatics and Data Science Research Fellowship. And I'll just share the link in the chat box. Yes, so here you go. Now, when you scroll down, you will find the program overview that this research fellowship program has been specifically designed for young researchers and students so that they can take advantage of bioinformatics resources so as to analyze the complex big data and become versed in bioinformatics. Now, when you'll scroll down, so you will find the proper full framework of the research fellowship program, which you will go through in the duration of your research fellowship program. So the first thing which you learn is about the introduction to different courseworks related to bioinformatics and computational biology, different NGS data types like genomics, transcriptomics, metagenomics, and epigenomics. Then the second thing is about the evaluation. So you'll work on an independent project topic selection through literature review. Then once your project title, your research question is selected, you will practically apply that. You will practically learn that using some example research projects which we have on our portal related to different domains like cancer, neurosciences, agriculture, biotechnology, and infectious diseases, and extract some meaningful insights from those large data sets. Now, next is about the comprehension and analysis, where you'll do some exploratory analysis. Then after that, you will work on investigation, and whatever you have gone through, in your investigation, your learning part, your analysis part, you will prepare a scientific report out of it and submit a project. So this was a brief about the framework of the research fellowship program. Now, if you scroll down, you will find three different fellowship lengths. That is, one is of 45 days, another is of 60 days, and then is of 90 days. And the pricing for 45 days in dollars is $350. For 60 days is $450, and for 90 days it's $550. Those who are in different location, for example, those who are in Africa or Nigeria or in Brazil or in India, so they can choose accordingly. They can choose the currency accordingly, and the pricing will then show according to their location. So right now the pricing in is USD, so it's reflecting in dollars. If you choose according to your location, it will start reflecting according to your currency. Now, if I talk about the resources, so the common resources which you have access, which you'll avail once you'll enroll in this program is, you will have access to our OmicsLogic Learn portal, where there are multiple courseworks, example research project, and case studies through which you will gain conceptual knowledge. You will have access to our T-BioInfo Server, which is an AI-based cloud pipeline having different toolkits performing different functionality. Then you have access to our coding sessions, our session recordings, online mentor support through Skype groups, online technical support, and certification after the completion of the program. Now, these were the common resources. Now, what's the difference between the three is, in 45 days tenure, you will be having access to the common group discussions and the online mentor support. But if you opt for 60 days and 90 days, you will have an opportunity to to have one-on-one mentor guidance as well related to your research project, your queries, or for future career guidance as well. Now, how to proceed with the payment checkout. For example, if you're interested in 2 months research fellowship, you can click on buy now. It will redirect you to the OmicsLogic checkout page. If there are any coupon codes or available, you can you can paste the coupon code here. You can add your user details your country, your occupation, your organization, and then click on this checkbox to proceed with the payment checkout. You can pay via Stripe, PayPal, etc. So this was brief about the registration part. Now we have scholarship also. So this is the scholarship form. Whosoever like they're facing any financial problem or something, so they can fill out the scholarship form, and we'll be texting or mailing you back your after reviewing your scholarship form. For further queries, you can mail us at marketing@omicslogic.com, and the official number, the Indian official number is 9876134120, and this number is active on WhatsApp also. So feel free to reach out us at marketing@omicslogic.com for further queries. And now I'll just stop sharing my screen, and let me know if anyone is having any doubt or queries related to registration part. Thank you. So I guess there are no more questions, and then we are good to go. And thank you, Dr. Raghavendran and Ojasvi and Sanalika and everyone else for being a part of this webinar. So take care, and please share your queries if anyone is having on marketing@omicslogic.com. Thank you.